Okay, so this is part 2 of chapter 2. We're going to move on with selective and differential media. We're going to kind of compare the two of them because they are very useful in the isolation and identification of specific microbes. So a medium can both be selective and differential. An example is McConkey agar. I may ask you to give me an example of a selective and differential media, and that answer that I'd be looking for is McConkey. But what it does is suppresses the growth of some organisms while producing a visual distinction between the ones that do grow. So it will keep the background organisms, normal flora, non-disease causing flora. It'll keep them from growing, and then the stuff that does grow, it will look different. So E. coli is going to look different from Staph aureus, which is going to look different from Pseudomonas. So basically dyes are used as differential agents because many of these dyes that are put into these plates, these agar media, are pH indicators. And that change in color in response to the production of acid or a base will help us to be able to identify between multiple organisms. So we also have a couple of miscellaneous types of media that we use. One is reducing medium. What this does is contains a substance that absorbs the oxygen or slows the penetration of oxygen. And reducing medium is very important in the growing of anaerobic bacteria. Anaerobic, implying that it does not like or grows better without oxygen. It does not like oxygen. It grows better without oxygen. Anaerobic. And that is a test question. Transport media used to maintain and preserve specimens that have to be held for a period of time before clinical analysis. And part of the issue with getting testing done for COVID-19 is that there was they had to have all this transport media because the little, depending on what test they did, if they did the nasal swab, the nasopharyngeal swab, they had to preserve it in a little bit of liquid medium, and then put it in. It had to be in a vial that closed, and then they would have to ship it, and that would assure that the specimen was viable when it got to the lab. And there was a bit of a shortage of those vials and the transport media, which kind of caused a little bit of a delay initially in the testing. Miscellaneous media, more miscellaneous media, carbohydrate fermentation media. So what this has is sugar. It contains sugars that can be fermented by certain organisms. And there's also a pH indicator that shows this reaction. So microorganisms that have the ability to ferment carbohydrates or sugars then will show a difference in color, and that will show these different results. If specimen B is known to cause this color change, then we know that this would be specimen B and this would not be specimen B. So examples here are it can make it look it can make it alkaline, it can make it acidic, and then it can make acidic with gas, and you can see this little bubble. So all these results are indicative of different types of microorganisms. And basically we just use a flow chart to say, okay, is it doing this? Yes. Then we go on to the next test. Is it doing this? Yes. Go on to the next test, and then we get all the way to the bottom where we are able to identify that organism. There's also assay media and enumeration media. Assay media is used by technologists to test the effectiveness of antimicrobial drugs. It's used by drug manufacturers to test disinfectants, antiseptics, cosmetics, and so on. Enumeration media is used by industrial and environmental biologists to count the numbers of organisms in milk, water, food, soil, and other samples. One example of this in the hospital is that we would have to, I forget, I think it was the water used, something used for, I'm going to totally draw a blank here. Dialysis machines. And we would have to check that water here and there. I believe it was from the dialysis machines. And then there was also a filtration unit in the OR that we would have to incubate the specimen with this enumeration media periodically to verify that the filtration systems were working properly. All right, so moving on to the next step. So that was all about inoculation. Now we're talking about isolation. Isolation is based on the concept that if an individual bacterial cell is separated from other cells on a nutrient surface, it will produce a discrete mound of cells called a colony. Now I mentioned colony earlier, so when we're talking about macroscopic morphology, we're talking about what we can see with the naked eye on a plate or in a test tube. And what grows on those plates, what grows on the agar on those plates, are referred to as individual colonies. So a colony is a macroscopic cluster of cells appearing on a solid medium arising from the multiplication of a single cell. What does it require? Isolation requires a medium with a firm surface, a petri dish, and an inoculating loop, which is a little metal wire with a little loop on the end. So, this is just showing, we have a mixture of sample of cells. We culture the plate and you can see individual colonies of both types is basically what this is saying. Other methods for isolating bacteria, I'm not going to worry too much about this, but these are just examples of how you can isolate bacteria. All right, so now we have moved on to inspection and identification. And I guess I kind of, isolation, yep, okay. So as I said before, inspection and identification many times are kind of grouped together. The microbes can be identified through microscopic appearance. We can look at it under the microscope. We can identify microbes through characterization of cellular metabolism based on what it does to the plates, what it does to the media in the tubes. We can determine nutrient requirements, products given off during the growth, presence of enzymes, and mechanisms for deriving energy. And all of these things might seem foreign, but the fact of the matter is like I said, it's like a flow chart. We get a throat culture, we automatically put it on 2 specific plates or 3 specific plates, and based on that information, it will lead us to the next test. If it's positive on this plate, negative on that plate, then we go, exit stage left and, do test A, B, and C. And if it's, positive on, test A, but negative on the other 2, then we'll, we might be able to derive a definitive identification based on that, or we might need to go further and test more. In addition, ways that we can identify microbes is through genetic and immunological characteristics through specific testing, as I mentioned before. So microbes vary in size. Yeasts are usually 3 to 4 microns or micrometers, and the smallest bacteria measure around 200 nanometers, and the largest is about 750 micrometers. Viruses are way smaller than bacteria. Yeasts And yeasts are usually a little bigger, and then, I mean, you're going to see some pictures here. I think it's in your book. Yeah, so here's the picture. So you can see bread mold at the top, and you can see a hydrogen atom at the bottom, and we know that hydrogen atoms are very, very small. You can see that E. coli kind of falls right around the middle. Red blood cells are bigger than those. HIV is smaller than E. coli. When you look at the individual sizing, these aren't necessarily to scale when you start looking at the numbers here. But just to give you a visualization of the difference in sizes. So I'm going to stop here, and I'll pick up on the microscope in part 3 of chapter 2.